The next speaker is none other than Dr. Jaime Yassif from the Nuclear Threat Initiative. And she's going to be talking to us about strengthening governance of dual-use bioscience research. So Dr. Jaime Yassif is a senior fellow for global biological policy and programs at Nuclear Threat Initiative. She is currently leading several major biosecurity projects with a particular focus on strengthening bioscience governance and reducing global catastrophic biological risks. Dr. Yassif previously served as the program officer at the Open Philanthropy Project, where she led the Biosecurity and Pandemic Preparedness Initiative. In this role, she recommended and managed approximately $40 million in biosecurity grants, which rebuilt the field and supported work in several key areas, including developing new biosecurity programming at several leading think tanks, establishing the Global Health Security Index, initiating new biosecurity work in China and India, and framing a new public discourse about global catastrophic biological risks. Prior to this, she was a science and technology policy advisor at the US Department of Defense, where she focused on oversight of the Cooperative Threat Reduction Program and East Asia security issues. During this period, she also worked on the Global Health Security Agenda at the Department of Health and Human Services, where she helped lay the groundwork for WHO joint external evaluations and the Global Health Security Agenda Steering Team. She holds a biophysics PhD from UC Berkeley and an MA in Science and Security from King's College London, War Studies Department, and a BA in Biology from Swarthmore College. Please join me to welcome Dr. Jaime Yassif.
I'm delighted to have the opportunity to join these really important and valuable conversations that are taking place today and over the next few days, and really grateful to the organizers for including me in these discussions. So it's great to see you all today. I'm also really sorry I couldn't be there in person. I am dialing in today from Cambridge in England. And it's great to see a lot of friendly faces and new friends. So I'll be talking about strengthening governance of dual-use bioscience research and continuing our thread of discussions about how we can build a safer world and how we can really protect against biosecurity risks. And it's really great to have the slot immediately after Megan Palmer, because there's a lot of common ideas and shared perspectives. And so I think these will go well together, these ideas. So I'm going to start by talking at a very high level about some of the risks as we see them at NTI, and then shift to some proposals for how to address these risks, and specifically some of the concrete actions that NTI is taking to reduce biological risks associated with rapid advances in technology. So first of all, at a really basic level, I think this audience and the synthetic biology community understands that rapid advances in biotechnology and technology across the board offer tremendous opportunities for public health, agriculture, economic development, and a number of other areas. But in order to really ensure that these advances can continue to flourish, we also need to safeguard these tools and these technologies, because they do pose risks of exploitation. And I'm going to talk a little bit more about that as we progress through the talk. But at a basic level, as Megan said a few minutes ago, one of the key drivers of the changing dynamics in this community is that it's easier now to read, write, and edit DNA and RNA, which is the blueprint for all living organisms. As you can see in the cost curves on the right that were developed by Rob Carlson, it's been getting exponentially cheaper in recent decades to read, write, and edit DNA. And I think this community knows this as well as anyone does. So what does this mean in practice for our ability to manipulate biological organisms? So first of all, it's easier to generate pathogens de novo or from scratch. And it's also easier to engineer them. You can make them more virulent. You can make them more transmissible among human or animal populations. And it's easier to make them less susceptible to medical countermeasures like vaccines and other therapeutics. And in fact, this kind of research is underway. There are legitimate researchers that are doing these kinds of modifications and these kinds of engineering tasks. And most notably, a recent example from a few years ago was when the laboratory in Canada basically synthesized a horsepox virus from scratch using $100,000 in mail order DNA. And then they subsequently published the paper that include all the methods for how they did this. And this was very controversial at the time. And I think different parts of the biosecurity and bioscience research communities have different views about the costs and benefits of taking on and publishing a project like this. And there are many other examples if you look further back. And I just sharing these snapshots of various news pieces and opinion pieces that have came out in recent years to show that this is a very live topic for the biosecurity, for the security community generally, for the biosecurity community, and also in public discourse, asking real questions about what is safe to do in synthetic biology, what is crossing the line, how do we balance our needs to advance science while also making sure that we safeguard this technology and we don't inadvertently create a future where we face catastrophic biological risks. And I want to sort of drill down a little bit more when I say catastrophic biological risks. I want to be very clear. And so, as I said before, it's incredibly valuable that there's a democratized access to a wider range of tools in synthetic biology and biology across the board to engineer biological systems. But what that means in practice is that there's also democratized access to a wider range of actors to engineer pathogens. And unfortunately, these do pose risks. And so as we see it, we believe that there's an increased risk of non-state actor attack with a biological weapon, and perhaps one that is more sophisticated. And based on recent history, we know that there have been non-state actors that have been interested in biological weapons. And so the intention and the threat is real, and we have to worry about maybe something like that's more serious could happen in the future. That is definitely a distinct possibility that we should guard against, in my view. We also see that there's an increased risk of accidental release with engineered pathogens. So if you look at the data, accidents do happen. Humans are fallible, equipment is fallible, and mistakes are made. And there have been examples of laboratories around the world accidentally releasing materials that they've been working on, and to the extent that that those laboratories are working with pathogens that are engineered, the consequences could be severe. And so again, we need to be thinking about how do we reduce those risks. And finally, this last point is a little bit speculative and more uncertain, but there's a possibility that these evolving capabilities could possibly shape the risk of state bioweapons development in the future. And so in this backdrop where you have all these developments, both beneficial and with risks, my bottom line here for this slide is that governments can't keep up. They are, our Global Health Security Index, which NTI developed in partnership with the Johns Hopkins Center for Health Security and working in close collaboration with the Economist Intelligence Unit, we published an index back in 2019, and a key takeaway from that index is that fewer than 5% of countries around the world provide oversight for dual-use research of concern, including with pathogens of pandemic potential. And so while governments and national academies have done extremely valuable and important work to sort of increase oversight and governance of this important space, it has been too slow and it's not enough. And I've shared a few examples here in the bullet points below of valuable, important work that academies and governments have done. And I'm happy to discuss that in greater detail at another time if there's an opportunity and interest. So what do we do about this? I think there are two really big risks that we want to guard against. We want to, protect the legitimate global bioscience research and development enterprise because it's so valuable to all of us. And we want to ensure that it's not exploited by bad actors who might be seeking to use it for weapons development purposes. We also really want to reduce the risk of some sort of laboratory accident that could release engineered pathogens into the environment, endangering countries around the world and the entire global community, and potentially creating catastrophic consequences. So those are the two big goals in my view. But how do we do this? And I would argue that there's no silver bullet here. There's no single intervention that will take risk down to zero. That's just not a reality. And so what we need is a layered defense. How do we do this? So there are multiple intervention points throughout the bioscience research, development, and commercialization life cycle. And each of these points is an opportunity for a different group or organization to meaningfully reduce risk. So it starts at the project conceptualization and funding stage. Funders can do more effective biosecurity review before they decide whether to award a grant or a contract. There can be more effective oversight as work is progressing in academic and industrial research facilities. We can have more robust oversight from a biosecurity and a biosafety perspective. Providers of materials and services, for example, DNA synthesis screening companies do have a tool at their disposal. By screening the orders, they can ensure that the building blocks of potentially dangerous pathogens don't fall into the wrong hands. And I think James Diggins' talk was an excellent example of Twist's work in that area, which has really been leading the field. And then finally, publishers also have an opportunity to review papers before they're published to make sure that it doesn't contain what we sometimes refer to as information hazards. And so I won't get a lot into DNA synthesis screening because I think James did a great job covering it, but I'll just share a few quick points. So, as we know, it's getting faster and cheaper and easier, but DNA synthesis screening is voluntary. There's no country anywhere in the world that requires it. All the providers that are currently screening are doing so voluntarily, and that's an estimated 80% of global market share. So not everyone screens, and that's a significant gap and a vulnerability. And what we're seeing already is that benchtop synthesis devices are coming online even now, and that they will continue to evolve their capabilities in the coming years. And that there's a significant chance that part of the market will become decentralized, and we have to figure out how do we safeguard and screen orders on benchtop devices to make sure that there's not a huge gap in the system. So to recognition of these challenges and these risks, NTI started the Biosecurity Innovation and Risk Reduction Initiative back in 2018, recognizing that these advances are moving faster than governments can keep up to provide effective oversight, and reflecting our desire to take action, engaging both the public and the private sector to develop concrete tools that can really meaningfully reduce risks. And so here are some, just to give you a sense of the flavor of some of these meetings. We've had a number of discussions with some important policy and thought leaders in recent years that have contributed really valuable insights as we've worked to build up this initiative. And a key milestone for us was in January of 2020. So NTI partnered with the World Economic Forum to publish a report on biosecurity, innovation, and risk reduction. And the central focus of the report was really about taking a global approach to DNA synthesis screening and thinking broadly about global governance of dual-use bioscience research. And two of the key recommendations we came out with were, one, to establish a technical consortium that could develop an international tool or common mechanism for DNA synthesis screening, so we can go from 80% of global market share much closer to 100%. In terms of screening, and to establish an international entity that's really dedicated to biosecurity risk reduction to sort of tackle this as a global problem and help countries and all the different sectors that are involved work together to really create a comprehensive, effective approach to risk mitigation. So we've been really focused on this as a very high priority for us on the bio team at NTI, and we launched the technical consortium back in May of 2020. Twist, Bioscience, and James Diggins are engaged in it, as are a number of other key members in the DNA synthesis provider community and industry, and really delighted to have their participation and engagement. And we are also working with international partners to scope out what a new international biosecurity entity might look like. We're working to shape the mission and scope and to work to build international support for that effort. Here's a quick slide to sort of give you a quick overview of what the common mechanism involves. In the interest of time, I'll be really brief. Suffice it to say that it includes both customer screening as well as screening of orders, and there's a whole sequence of decisions and decision support tools that we are developing in partnership with industry to develop this common mechanism, and we're building it based on best practices that industry has already developed and trying to take it one step further. I'm happy to discuss that in more detail at another time. And I did want to take the last minute or so to just talk about this new biosecurity entity that we're working on. And we envision that this new international biosecurity entity would be the home for this common mechanism for DNA synthesis screening, but it would have a much wider mission. And so the mission, as we've been shaping it, is to reduce the risks of catastrophic consequences due to accidents or inadvertent use or deliberate abuse of bioscience and biotechnology. And the way that we're looking to do this is by promoting stronger global biosecurity norms, and to develop tools and incentives to support adherence to those norms so we can work together to meaningfully reduce risks. It's going to focus initially on DNA synthesis screening, but over time, our understanding is that it can encompass the broader suite of intervention points that I was talking about earlier to really make a meaningful layered defense against these risks. My final point is that we envision this entity will be globally oriented, agile and innovative, collaborative and transparent, and free to engage with a diverse range of stakeholders, because this is a complex problem that requires all hands on deck. So thank you so much for your attention. It's been a pleasure to join you today. And there's more information about our program and our team on the NTI Bio website. And with that, I yield.